Yeah, so I'm not sure what happened, but let me welcome all of you. It's terrific that everybody could make it. We have altogether at this point, I think about 120 people signed up for this. And it's tremendous that almost 60 people so far managed to join this meeting. So as James said, we want to start off, James and I, giving just a very brief introduction to this. And we want to spend the bulk of the meeting hearing from everybody. Now, with this many people on the call, one sort of might question the wisdom of having everybody say something about themselves and what they think they can contribute to this working group. But this might be the only time, really, or one of the very few times that everybody can kind of get together in an interactive way. And so we figured that that would be a good way to spend time. There are really two key objectives in this meeting. One is to discuss the objectives of the working group, and the other one is to determine, to some extent, or get input, at least initial input, on what the subgroups of this working group will be, and ideally who will be able to lead subgroups. So let me maybe move ahead to the James is back. So James, you can go over on the next slide.
The critical web failure. I don't know what happened. Thank you for your patience. So to introduce ourselves, I'm James Glazier, PhD, professor in Intelligent Systems Engineering in the Biocomplexity Institute at Indiana University, Bloomington. My area is multi-scale modeling and software infrastructure and model sharing. And I'm one of the co-leads of this project. Reinhard?
I'm Reinhard Laubenbacher. I work in, I'm a mathematician by training, and I work in multi-scale modeling of respiratory diseases, among other things.
Jim?
I am Jim Sluka. I'm a computational biologist and a computational chemist, and I'm kind of a support person for this group.
I'm Bruce Shapiro, and I'm a mathematician, meteorologist, and a project manager. I've got 23 years in the Air Force and 8 years of project management for electronic systems acquisition for the Air Force also.
Okay. So I wanted to give you just a little bit of history about this. I had started discussions back in the spring with Grace Peng about whether there should be a working group under IMAG MSM on the generic topic of viruses and immune system. Erica Mohan at Carlow University and I put together a proposal initially in June. We sent that in for review by the IMAG MSM steering committee. They suggested adding a co-lead, and Reinhard Laubenbacher was generous enough to join us as co-lead. The revised proposal was submitted in July and approved on July 23rd, at least in preliminary form. And so this is our first formal meeting of the working group. Bruce, if you can make it. One of the things that I've been asked to do by Grace is to remind you that this is not what was said here, and what is posted on the working group site represents the work of individual scientists and not the opinion of NIH. Since this is an area of some interest, both politically and socially, we have to be extra careful about that. If you are interested in looking at the web page, you're welcome. Suggestions for improvements to the web page and the mission statement are welcome. The main purpose that we developed for this idea is that while there has been a lot of work on epidemiological modeling of viruses and pandemics, while there's certainly very substantial work on in-host responses, modeling the immune system and in-host responses specifically have been addressed much less. And that seemed very properly to be the focus area for IMAG MSM, whose remit is to provide basic biological understanding and medical insight using multi-scale modeling. And suggestions for improvements to the webpage would be much appreciated. Okay. From there, we can enunciate perhaps a very high-level dream of what one might want to do with this. Fundamentally, one can imagine building a integrated multi-scale representation of infection and the body's response to it, and using that to understand the basic biology of immunology, predict the course of infection in individuals, the immune response, and potentially to optimize and develop new therapies. We're talking about using multi-scale modeling, as always, in IMAG MSM for the integration of very heterogeneous data sets, multiple scales and types of models, and then their use to understand not only human biology and medicine, but also its relationship to what's observed in the laboratory using model animals, and in the case of viral infections, or also bacterial infections, particularly their behavior in host organisms in the wild. We are certainly not a COVID modeling group, but of course, the experience with COVID will motivate our ideas and focus, perhaps, to begin with. And so thinking of COVID as a disease that came from bats, it's quite interesting to think about the differences, for example, between bat immunology and human. And so that is perhaps an appropriate area of consideration. Ultimately, since this is a cross-agency initiative, we're interested in biological knowledge of all kinds, basic biology, and then on the medical side, in improvements of diagnosis, prognosis, and therapy optimization. This is a dream, and this is a working group. This is not a group that does, as a working group, we discuss and we make recommendations about planning. The working group itself does not do modeling. It's the people in the working group who do modeling. But I think it is helpful to have a vision of what might be possible if we had perfect models, to think about how to organize what we might want to do. And that's my opinion. We're very much open to alternative points of view. Again, we've specifically focused on in-host, but considerations of how in-host models might connect to population epidemiological models or infection models are also, of course, interesting. And then I'll turn it over to Reinhardt, who will talk about short-term deliverables and run the rest of the meeting.
Yeah, thank you. James, what I want to talk about is our short-term goals, and here's a slide, and there are more specific goals in the document. So I don't want to read the slide to you. You can read it yourself, but if I were to paraphrase what I hope would come out of this in the short term, meaning maybe on the order of 6 months or so, is really a blueprint, and that can take the form of one document, an article, 2 articles, whatever, but a blueprint that of how multi-scale modeling can contribute to in-host modeling of viral pandemics as a tool to deal with the next pandemic that comes along. And put it in a format that for example, an NIH program officer can pick up and say, you know, this could be the basis of an RFA, or a researcher could pick up and say, you know, here I see a piece of how my work would fit into this, or a graduate student could pick it up and say, you know, this is great. I think I want to contribute to this part, and I can see the bigger picture. So that's really my vision. And of course, this is a volunteer group of 100 people, and you know, ultimately what the end result will be is hard to say because people self-organize. But I do hope that at least in the short term, that is a goal we can accomplish. And what happens in the long term, we'll just have to see. I'm sure whatever happens, great things will come out of this. So if you go to the next slide, Bruce, there are a number of action items that we need to deal with, not now, but I think as we go along. And here are some items. We have created a Google document that people can add comments, suggestions into, and participate in the discussion. Here is the link So please put your comments in to the extent that you cannot voice them here due to time constraints or think of them later. This also will go to people who can't participate in this meeting. Next item. And so there are some more things. One thing I want to mention is you've all registered for this working group, but there is another registration needed, and that's for the IMAG MSM website, at least if you want to post to the website. This registration process is non-trivial. A PhD may or may not be sufficient to navigate it, but we have detailed instructions on one of these slides. So, and if you have any problems, contact Jim Sluka or any one of us. So here it is. And that's really how far I want to take this in terms of describing what we think this working group is. And I think we should, to the extent that we can, really take the whole rest of the time to hear from you. Now, there are at the moment 72 participants on this call, so I'm not sure how well we're going to do. What I hope is that each participant will be able to take 1 minute or less and tell us who you are, what you work on, and what you would like to contribute to this working group. In particular, which subgroups you might want to be interested in. So here's a list, a tentative list of subgroups. It's not exhaustive. If you have other ideas for which subgroups we should have, and in particular, if you're interested in leading a subgroup, then please say so. Without further ado, just logistically, Bruce will call on each of you as we go along. If you prefer not to say anything, that's okay, but if you do, please stay in that time limit so that everybody has an opportunity to speak. So Bruce, you want to get started?
Okay, I'll just get started. I'm going to go down the list of everybody that signed up, so there will be some that won't say anything. So Dr. Yang, Dr. Osborne, Dr. Moore, Professor Moore, Professor Bueller.
Hello, hi, yeah, I'm Marcus Bueller from MIT here. I work on protein modeling, multi-scale modeling, especially protein materials, and including recently some modeling on COVID pathogen mechanics. And this working group is intriguing. I've been involved in other multi-scale aspects with IMAG, with Grace over the years, and I'm very excited to be part of this group and contributing in any way I can. Thank you for leading the effort.
Thank you. Dr. Baat, Dr. James Clonig, Mr. Gary Ahn, John Rice.
Hello, my name is John Rice, and 40 years in Department of Defense modeling, simulation, training, operational systems tests, stuff like that. Retired, drifted over, became a simulator as a standardized patient at a medical school, got really interested in the applications of modeling, simulation in healthcare, ended up drifting over to NIH and discovering the multi-scale modeling consortium, got involved with the Medical Device Innovation Consortium, which took me into the FDA, modeling for clinical trials. Also, for the last 4 years, been a member participant of the Federal Pandemic Prediction and Forecast Working Group, which was originally chartered out of the Office of Science and Technology, and then discovered bats related to COVID virus, and talked to James. And I am really excited to see this working group.
Thank you. Dr. Liu. Professor Liu.
Hi, this is Yang Lu from Lehigh University. So I think I'm more in a smaller scale. So I work mostly from the subcellular scale to cellular scale. So in the smaller scale, I work on using molecular dynamics simulation to predict the binding between RBD binding site and ACE2, and also trying to figure out whether there is a potential drug that can inhibit the binding behavior. I also do a little bit more on a cellular scale, mostly study the virus transport in the droplet, in particular for the airborne transportation process. So I'm very excited to see this school and be willing to contribute in any way.
Thank you. Thank you. Dr. Macklin.
Hi, I'm Paul Macklin. I'm at Indiana University. We have been leading a multi-institution coalition to develop an in-host model that goes from viral replication scale, connects up to spread of plaques within a tissue, immune integration, and spatially resolved immune modeling in that tissue scale model, then integrating that to a systems level ODE model of immune expansion, and been very grateful to lead a wonderful coalition of people, most of whom are on this call today. And I'm very excited to see that integrate with other work here. And I would personally like to see a good baseline immune model that comes out of this that we can apply to other disease and physiologic processes. We're already looking at using it for melanoma and immuno-oncology. So very excited to be here. Thank you. And pitch in wherever you think it makes most sense.
Thank you. Thank you. Professor de Almeida.
Hello, I am Rita de Almeida from Porto Alegre, Brazil, from the Federal University in Rio Grande do Sul, which is the southernmost state in Brazil. I mostly work with either compost cell simulations of crawling of cells, but also with the analysis of gene expression. And we have with James a preprint on the dynamics of the immune response for the SARS-CoV-1. And we are presently also applying the method for CoV-2 and comparing with CoV-1 data. All these data we have got from either from public databases or from collaborations, but we can try to give some numbers of how the system works in this level of expression and gene expression and everything. So that's it.
Thank you.
And I'm very excited. Thank you.
Thank you. Dr. Gevert.
Hi, I'm at Stanford University. I'm in the Department of Medicine and Biomedical Informatics Research. We focus on multimodal data fusion, linking clinical and EHR data with molecular multi-omics data and with imaging data. I would be happy to be involved in any working groups that focus on integration within and across scales. And we have some preliminary work that we did on COVID patients using lung CT imaging data.
Thank you. Dr. Fader.
Hi, I'm Jim Fader. I'm at the University of Pittsburgh. My group is interested primarily in studying intracellular signaling dynamics, but we've gotten into modeling viral replication and viral life cycle as part of, oh my goodness, Paul Macklin's consortium. And so, you know, have a lot of interest, particularly at the subcellular level. We also develop modeling tools for representing models of intracellular signaling that could be scaled up to make multi-scale models, and it would be really fun to discuss issues of language development and simulator development technologies with various people in this working group. Thank you.
Thank you. Professor Tawahi.
Hi, Maren Tawhai from Auckland Bioengineering Institute at the University of Auckland, New Zealand. My main focus is on multi-scale models of respiratory system function, so integrated models of soft tissue mechanics, so soft tissue deformation of lung, integrated with ventilation distribution, models for perfusion distribution, gas exchange, particle and inhaled gas transport. So really our work provides the link between the patient, CT imaging and function, So more recently, we've started looking at lymphatic function as well. So looking at the emergence of dysfunction when there's systemic inflammation or, localized injury. So acute respiratory distress syndrome sort of emerging as a consequence. So very interested in contributing to tool frameworks for simulating respiratory function. So frameworks that others can use to complement this research.
Thank you.
Bruce,
Dr. Smith.
I'm sorry, could we go to Bruce Lee, who needs to drop off in just a little bit?
Oh, yes.
Okay, hello everyone. Thanks, Bruce Lee. I'm a professor at the City University of New York. I've been an investigator and member of the MIDAS network since 2007, so almost 15 years. We were embedded in Health and Human Services during 2009 H1N1 pandemic, and we've basically been involved with every national response and are currently working with CDC and other folks in terms of the COVID-19. So I thought this would be an opportunity to sort of see, we could share some of our experiences with the MIDAS network and those things like that, and then see what types of cross bridges there might be between the efforts here and ongoing efforts here and the global efforts. We're also working with WHO, World Health Organization, and Gates Foundation and other entities. And we do various types of models, including very detailed agent-based modeling. They're multi-scale.
Okay, thank you. Now, Dr. Smith, Amber Smith.
Hi, I'm Amber Smith at the University of Tennessee Health Science Center. My group is mostly interested in modeling and validating immune response models to viral and bacterial dynamics. We use a lot of mouse models and patient data to try to understand the immune regulation. And I am also involved with the group that Paul Macklin had discussed earlier.
Thank you. Dr. Drart. Dr. Yi, Keming Lee Yi.
Sorry. So this is Kaiming Yi, the professor, department chair in the Binghamton University. Actually, I'm so glad to see the formation on this working groups. Actually, Grace and I, we start this multi-scale mathematical consortiums back to the day I was in the NSF, National Science Foundation. Actually, she and I, we jointly funded the first batch of the projects under these consortiums. My research is more focused on infectious diseases. We work on a lot of the vaccine developments, influenza virus and vaccine developments, cancer, and related to the COVID-19. We work on the COVID vaccine development and also on the SARS-CoV-2 disinfection technologies. And we're happy to work with everybody on to see the growth of this consortium.
Thank you. Dr. Stratford.
Yes, hi everyone. I'm Bob Stratford, and I'm the director of the Modeling and Simulation Program at Indiana University School of Medicine. That program is housed within the Division of Clinical Pharmacology. So in clinical pharmacology, we're interested in the intersection, if you will, between human physiology and physical chemical properties of drugs. And we use models to integrate those two fields. Virology is new to me, so I'm excited to be a part of all of this, to learn about virology, and feel particularly well suited to participate on the therapeutics and medicine subgroup. Thanks.
Thank you. Dr. Kavridakis. Professor Karniadakis.
Hi, I'm George Karniadakis from Brown University. I've been with the program, the MSM program, since 2003. And my interest here, I think actually this is a great idea that you guys had, and I totally endorse it. You should have done this more than 10 years ago, then we had really a big impact now. But what I am interested in is integrating machine learning tools and the multi-scale modeling. And in particular, I like the way you have divided in these topics, including diseases, diabetes, for example, which is very important here. But also, I would like to add to that list, I would like to add sickle cell disease, because that, of course, addresses a very vulnerable population group right now, African Americans, and so that I would like to add more diseases, just not just diabetes.
Thank you. Dr. Cool.
Yeah, hi. I'm actually more interested in the larger scales. I also, like George, do multi-scale modeling and data-driven modeling, physics-based learning. And we're interested in learning on the larger scales, more organ, all the way up to population level. And I'd be happy to lead one of these subgroups that are more on the right lower side of the sheet that you're projecting right now. Thanks.
I want to emphasize that this list was very provisional, and we've heard people doing molecular modeling, we've heard people doing airway modeling and potential interaction with ventilators, and of course connecting up to higher scales also is of great importance. So please don't, and we heard from Amber Smith about working on bacterial co-infection. So the goal of this list was to provide an initial set of talking points. Please don't feel that we're trying to exclude anyone or any set of ideas because it's not a bullet point on this list. One of the reasons we're going through the room is because we want to make sure that everyone's ideas are represented.
Thank you. Dr. Okchipiniti. Dr. Mapter.
Yeah, hi, I'm at Indiana University School of Medicine, and I'm in the modeling and simulation group, and I've got a training of theoretical biophysics, but I'm mostly interested in modeling infectious disease for last couple of years, and I'm working with James Glazier's group and also in Paul MacLean's consortium right now, and my main focus of the modeling is to construct a virtual patient population with adaptive and innate immune response with clinical data calibration with them mostly. And also we are doing physiologically based pharmacokinetic model for different drugs like antiviral or anti-inflammatory drugs together.
Thank you. Dr. Wang. Professor Dang.
I'm Ru Van Den from Stony Brook Applied Mathematics Department. We have been for the last approximately 10 years developing multi-scale algorithms for simulating activation and aggregation of platelets, joining with our biomedical engineering department. I'm coming here to learn if we can contribute our skills in applied math and computer science development of algorithms, particularly in this modeling technologies.
Thank you. As I go through this list, James posted in the chat. I don't know if everybody's monitoring the chat, but if anybody has time constraints, you're welcome to just jump in line. Professor Lin.
So my name is Guang Lin from Purdue University. So my group is a data-driven modeling and the model discovery using machine learning technique. So the goal is we try to using data-driven techniques to automatically learn the symbolic representations of the multi-scale models, and basically help us to gain like insight interpretations behind the data, try to really discover the physical laws instead of learning the black box models. So we have developed various kind of techniques to basically to do the integration for models across different scales. So I think I can contribute on the modeling technology subgroup. Thank you.
Thank you. Professor Com. Dr. Zarnitsia, Veronica?
Yes, Veronica Zarnitsina.
I'm at the Department of Microbiology and Immunology, Emory University School of Medicine. I'm a physicist by training who moved to the area of mathematical modeling and immunology. And I'm mainly interested in immune responses to influenza and other viruses, as well as in linking within and between host scales. Also, I'm currently doing some work on longevity of immune responses, both for T cells and antibodies. And for subgroups, I think I will be happy to be part of the adaptive immune responses and integration of the within host and population scales. So thank you for organizing this group. I'm very excited and happy to look forward to work with.
Thank you. Dr. Johansson. Dr. Siegel.
Hi, everyone. My name is TJ Siegel. I'm a second-year postdoc at the Biocomplexity Institute in Bloomington at Indiana University with James Glazier. I'm the lead developer of CompuCell 3D, and my background is primarily in applied mathematics and multi-scale computational modeling, both in biomechanics and cellular and tissue dynamics, as well as software and simulation development. My related work to this group, I work on building the multi-scale viral infection framework in CompuCell 3D. That's a lot of how James and a few others of us, how we find ourselves here with our related work. And so with that, I'm particularly interested in contributing to the physiological models of the innate and adaptive immune response, as well as some of the aspects of modeling technology.
Thank you. Dr. Rafailovich. Dr. Darkin.
Yes, my name is Chantal Darkin. I'm at the University of California in San Diego. I'm a professor here in the Division of Physiology, but I'm a mechanical engineer by training. My main interest has been in developing model of aerosol transport and deposition in the lung. I've used mainly CFD and also multi-scale modeling, which we are currently funded through the IMAG to do so. I also done human and animal studies, and partially I started that partially because of frustration of having the right data to construct and validate the models. So I see myself more like at a good position to be between the experimental data and validation of model. In terms of the subgroup of interest that are listed, I could see myself both in the individual organ system in terms of the lungs, or the modeling technology for the transport model in the lungs. And finally, another group that could work would be the experimental data for model construction and validation.
Thank you. Dr. May. Mr. Wang? Yafei Wang? Dr. Lynch?
This is Miranda Lynch. Is that the one you're looking for?
That's close enough.
Okay. I'm Miranda Lynch, and I'm at Hopman Woodward Medical Research Institute. I do a lot of work in structural biology modeling, including molecular dynamic simulation and protein-protein interaction modeling. I would be interested in things related to the therapeutics because of my structural biology emphasis.
Thank you. Dr. Nakfi? Dr.
Hello, I'm Razanaki from Superior University. Basically, I have a background of semantic technology and knowledge modeling, and previously worked with the drug-drug interaction models and P4 and P5 precision and personalized medicine. And I will see myself more with a technical side in the modeling technologies, the integration side, and I'm looking forward to it.
Thank you. Thank you. Dr. Madamanacci.
Hi, my name is Bobby Matamanchi. I'm a senior data lecturer in the School of Information at the University of Michigan. My research has been on computational pedagogies and specifically on training other STEM disciplines, especially biological scientists, on using multi-scale modeling. I've worked with the CompuCell group a lot in that area. So if there are training initiatives, I'd be eager to help or even lead.
Thank you. Dr. Parhak.
Yeah, that's me. I'm independent, unlike you. I work outside academia. I currently own the most validated diabetes model. I'm a population modeling. If anyone knows if diabetes models validated against more populations, please let me know. I'll add more populations. I also converted this model to be a COVID model, and I'm looking for actual people to actually collaborate with for interpreting some of the US data. If someone is interested, let's talk. The model is an ensemble model, meaning it integrates other models. So I'm interested very much in integration part.
Thank you. Dr. Venkatramana.
Hi, I'm Srini Venkatraman. I'm from the Biocomplexity Institute at University of Virginia, led by Chris Barrett, Madhu Marathe, and Stephen Eubank. I'm an engineer by training, and I currently work on multi-scale models at the population level, both agent-based models and metapopulation models to understand spread of diseases. So especially for COVID, we have been supporting federal, state, and local partners with like COVID forecasting and different intervention analysis. So I'm mostly here to listen and learn and hopefully understand how to integrate some of these state-of-the-art models of within host into population scales and maybe help with translating it to policy and practice.
Thank you. Bard Ehrmantraut. I see you have your hand up.
Yeah, sorry, I got to go very soon. Bart Ehrmantraut, Department of Math, University of Pittsburgh. I work in all sorts of areas of math biology, and I'd be most interested in, I've been sort of working a little bit with James Glazier, and TJ Sago, and Eric Emotion, who's also on this call. I'd be most interested in contributing to the physiological modeling, innate immune system, and things like that.
Thank you. Professor Harris.
Yeah, hi. So I'm Leonard Harris. I am an assistant professor in biomedical engineering at the University of Arkansas. I've got a background in stochastic simulations, in modeling of complex intracellular biochemical pathways. I used to work with Jim Fader as a postdoc. Hi, Jim. Nice to see you here. So we have similar backgrounds. More recently, I worked as a postdoc at Vanderbilt University in the Center for Cancer Systems Biology. I do a lot of cell-cell interaction models for tumors. And so I'm interested in, doing similar types of simulations, but in other areas. I'm also broadly interested in multi-scale modeling methods, techniques for integrating across scales, and model reproducibility and integration, that sort of thing.
Thank you.
Can I just jump in here? I have to go.
Yes, please.
Okay, thank you. I'm Joanna Wares from the University of Richmond, which is a liberal arts college, if you're not familiar with it. My research focuses on infectious disease spread in the hospital, but also some oncolytic virotherapy with a little bit of immune system modeling. But my real interests have to do with this social issues category that you have here. Health equity and training and outreach, and creating curricula for students to understand all of this that you're talking about. So I'd be happy to help with some of the population dynamics modeling, but also would be really interested in the social issues, social justice type issues that are arising. Thanks.
Thank you. Thank you. Dr. Chen Liao. Mr. Slim Faradi. Dr. Alexander Hoffman.
Hi there. Yeah, my name is Alex Hoffman. I'm at UCLA, professor of immunology and director of an institute for quantitative and computational biosciences that really faculty span from precision medicine to genomics to dynamical systems. My research interests are in dynamical modeling and experimentation and systems biology approach on the signaling networks that control responses by macrophages and by B cells in the germinal center. And our models are experimentally pretty deep validated and detailed with hundreds of components. Our particular expertise is in how inflammation affects these processes. And so I'm particularly interested in interacting with people at different scales on the topic of comorbidities.
Thank you. Thank you. Dr. Monk. Mr. Rodrigo Santibáñez.
Hi everyone, I am Rodrigo Santibáñez. I am working here in Chile as a system administrator of some servers, but my main interest is system modeling. My PhD thesis was about the modeling of bacterial metabolisms and gene and control of gene regulation. So my interest will be working in the modeling technology group as a scientific. Thanks.
Thank you. Dr. Ragamani. Greg Forrest.
Hello, my name is Greg Forrest at UNC Chapel Hill. I just sent a text to chat with everyone to explain what I do in case I didn't get called. I have to go in a few minutes. I've been working with the Moscow lung. I'm an applied mathematician. I do modeling of basically how viruses move in mucus in the lungs and what the transport properties of mucus are. I've been working with the Moscow Lung Institute for over 20 years to understand the pathologies of mucus in the lung. And for the last almost 10 years with Sam Lai in the School of Pharmacy, who designs and studies natural and monoclonal antibodies to fight viral infections in the lung or other organs. I work with other folks who have a chemical basis for trying to understand mucus pathology and how you might design drugs that would cross-link pathogens to mucus. So what I can bring to this group is I have a big modeling effort supported by NSF, by the way, but what I can do is I have a whole bunch of colleagues that are in clinical medicine. We have, lots of patients who have COVID, and we have lots of data for what the infection actually looks like and how it propagates and what the different trials are for treating the infection and for vaccines. So I can bring just that group of collaborators all across UNC.
Thank you. Professor Ragamani. Dr. Shoemaker.
Hello, sorry. Hi, Jason Shoemaker. A lot of familiar faces on this phone call. So I do computational modeling primarily of severe respiratory infection. So we do H1N1, H5N1, and of course COVID-19 in these days, mostly working at the physiological level. And that's really it.
Thank you. Mr. Carlos Lopez.
Hi, yes, my name is Carlos Lopez. I'm a assistant professor of biochemistry at Vanderbilt University. My lab is really working on modeling and simulation of complex network-driven processes in biology. So we're very interested in the dynamics of networks, how noise affects information flow in these kinds of networks. And then ultimately, we're very interested in understanding how data and models talk to each other. So we use a lot of high performance computing and Bayesian inference, Bayesian statistics to try to understand the relationship between models and data and really kind of bring together a model of math-based picture of biological processes. Thanks.
Thank you. Dr. Giori.
Yes, hi, my name is Ben Yuri, and I'm a research fellow at Harvard Medical School. And my research is funded on a number of ongoing DARPA projects and focuses on knowledge assembly, often from text mining, to build computational models of disease and therapies. And over the last 6 months, our group built a model that monitors the COVID-19 literature and automatically updates a model of that knowledge, and it then uses that to explain some experiments. And I hope to contribute to the therapeutics working group or also the modeling technologies group.
Thank you. Dr. Moore Siglio. Dr. Mochan.
Hi, I'm Erica Mochan. I am an assistant professor and program director for mathematics and data analytics at Carlow University, which is a small liberal arts college in Pittsburgh, Pennsylvania. And I work on ODE models of the intrahost immune response to virus and bacteria.
Thank you. Dr. Kim.
Hi, this is Eunyoung Kim at Infectious Disease at Northwestern University in Chicago. My research focuses on antiviral drug resistance and genetic variation in HIV host susceptibility to viral infection and disease progression, and also looking at the molecular evolution of the mechanistic change in HIV, SIV, and influenza, dengue, Zika, all the RNA virus in host defense. My study focused on systematically examine the host viral regulation interface to discover host gene and pathway involved in disease and whether they have genetic variation in host factors contribute to the individual difference in virus susceptibility. I'm using like a multi-center age cohort study, like well-defined sample obtained from those max cohort. I've been using next generation sequencing to look at their transcriptomics analysis and such. So I'm really happy to involved in this working group. Possible subgroup area could be viral life cycle and experimental data for modeling construction. Thank you.
Thank you. Dr. Velasco Hernandez.
Yes, my name is Jorge Velasco Hernandez. I'm a biologist by training, and now I'm professor in the Institute of Mathematics of the National University of Mexico. Our main research line has been the population dynamics of infectious diseases, modeling particularly on the tropical diseases, dengue, Chagas, also some respiratory infections. And from the beginning of this year, we've been involved in the modeling efforts for COVID-19. Informing the Mexico City government, and now we are dealing with the strategies for release of the vaccine, of the several vaccines that may be available to us. We have recently incorporated in my group people working in viral dynamics. So we have a grant also from the National University of Mexico on the population dynamics of COVID-19, and we would like to extend that to across scales and integrating viral dynamics. So I guess our interest will be mainly in this integration of within host population scales. And obviously, since we are in a country with severe limitations on resources and money and things like that, the social issues also is an important thing for us, particular things that have to do with equity and justice in the assignment of vaccine and other resources against this disease and others that may come. Thank you.
Thank you. Thank you. Damilola Olabode. Dr. Q. Christian Massa. Mr. Christian Kirachev.
Hello, can you hear me?
Can hear you.
My name is Chris, and I'm currently an EPSRC Doctoral Prize Research Associate at the Mathematical Institute in Oxford. My research focuses mainly on modeling filtration and decontamination of flue gas, and is in collaboration with W.L. Gore and Associates, known for Gore-Tex. I have extensive expertise in multiple scales modeling, and in particular using asymptotic analysis and the theory of homogenization, where one can systematically obtain upscale equations for complicated microstructures such as porous medium. I also have keen interest in mathematical biology, having worked on a few interesting projects at study groups, mainly on cell modeling and drug delivery. I also have expertise in reaction diffusion systems, moving boundary problems, and heat transfer. In terms of the proposed groups, I'm very interested in participating in the innate and adaptive immune responses, modeling of transport and other processes in the lungs, and any other general upscaling methodologies. If I'm to propose a subgroup, this would be on filtration or decontamination of surfaces, but I'm not sure this is within the scope of this work group. Thank you.
Thank you. Mr. Rahul Badani.
Hello, everyone. My name is Rahul Badani, and I am a PhD student in electrical and computer engineering at the University of Arizona. This is my terminal year, and I'm hoping to finish next year. I'm aspiring for a career in academia. My current area of the research is in computational biology with focus on dynamical systems and single cell data analysis. By training, I'm a computer engineer. And so mostly I will be interested in modeling technologies like using data to make models and doing predictions.
Thank you. Mr. Chris Kang. Mr. Mogre. Dr. Marcela Torres. Dr. Kupik. Dr. Heen. Dr. Craig.
Hi, my name is Morgan Craig. I'm a researcher at the Saint-Justine University Hospital in Montreal and an assistant professor at the University of Montreal. My group works on translational models of disease, including a variety of viruses and integrating therapeutics into that. So I'd be interested in contributing to the physiological models and therapeutics in medicine.
Thank you. Dr. Mahapatra.
Hi all, I'm Chittaranjan Mahapatra. So I'm a postdoc at Cardiovascular Research Institute, University of California, San Francisco. So my research includes the electrophysiological modeling, simulation of electrical activity in excitable cells in both at cellular level and network level. So I am here for collaboration for multi-scale modeling at organ levels. Thank you.
Thank you. Mr. Isaac Clapper.
Hello, this is Isaac Clapper. I'm at Temple University, and my interests have been in modeling of microbial communities, particularly in their environment, physical environment, mechanics and transport, chemical environment, metabolism, and ecological aspects. How all those things fit together and affect function of the community. Also with a little bit of host interaction work as well. Thank you.
Thank you. Professor Schnell. Paula Vasquez.
Hi, my name is Paula Vasquez. I'm a associate professor at the University of South Carolina in the math department, and I'm mainly interested in the transport processes on complex fluids. I actually work with Greg Forrest as part of my postdoc before with lung mucus, but in general, I work with complex fluids, both on the math side, but I also have a wet lab, so I always also work on the experimental side of things on microbiology. So I'm interested on both on the modeling technology group, part of the group, but also on the experimental data for model construction and constitutive equations, the formulation of constitutive equations. Thank you.
Thank you. Daniel Jonas. Professor Humphreys. Professor Gamel. Dr. Forgoston.
Hello, I am Eric Forgeston. I'm an applied mathematician who broadly works in the field of dynamical systems. I've worked in a number of areas within this field, but over the last decade or so, much of my work has been involved in population level spread of infectious disease, as well as transport within fluid flows. Much of this work involves stochastic dynamics, as well, more recently, machine learning and data-driven modeling. As far as this working group, I would be most interested in being part of the physiological models or some of the aspects of modeling technology.
Thank you. Surivdia Ayer Biswas. Dr. Miranda Lynch, I think we already had her. Dr. Tang.
Is that the Ting Ting Tang?
Yes.
Okay. Well, that's me. I'm at Ting Ting Tang, and I'm currently assistant professor of mathematics at SDSU, San Diego State University. So very grateful for this opportunity for interdisciplinary collaboration. Actually made possible because of COVID, which is funny. Personally, I have worked with both discrete time and continuous time models, basically ODE, PDE models, or matrix models, to model the population dynamic across different scales, like from basically cell to a population level. Recently, a little bit on meta population level as well. And so also developing numerical schemes to simulate and validate these models as well. So my work, I think, fit the best into the group of modeling technique, in particular integration within and across scale and integration between within host and the population scale. So in addition, I'm also actually very interested in basically connecting the two of the big list. One is experimental data for modeling construction and modeling technology. I was hoping eventually there will be a more systematic and robust strategies to basically funnel the data is available to actually all the advanced models that's out there in mass biology. And that's all. Thank you very much.
Thank you. Dr. Ford Versipt. Dr. Schwartz. Dr. Zillman. Anton Zillman.
Hi,
Dr. Donovan. Sorry.
I'm here. Yeah, I'm Anton Zillman from the physics department at the University of Toronto. We are currently mostly interested in cytokine signal integration, both on single cell level and also on the level of collective immune response. A separate but related issue, we've been working in last several years on kinetics of transport of various drug delivery vectors in blood and in tissue.
Thank you. Dr. Donovan. Professor Adler.
Good timing. I was just about to leave. Yes, I'm a mathematical modeler and mathematical ecologist at the University of Utah. I'm particularly interested in the interactions between immunology and epidemiology, sort of the multi-scale level. Within that, sort of an ecological side, I'm interested in virus-virus and virus-bacterial interactions and the role of evolution in the course of the pandemics.
Thank you. Dr. Gonzalez-Parra. Professor Chu.
Yes, I'm here. Tom Zhou, professor in mathematics and biomathematics at UCLA, trained as a physicist, and I'm mostly interested, mostly interested in statistical physics methods. So as such, I've worked in two areas that may be of relevance. One is the micro scale receptor engagement and viral entry, especially for HIV. And I'm also interested in testing statistics and data. So I'm here to learn. Thanks.
Thank you. Dr. Thompson. Mr. William Hart. Dr. Rachel Monspach. Dr. Diallo.
Yes, Mamadou Diallo. I am a program director at NSF in the division of chemical, bioengineering, and environmental transport systems. So I'm here to learn and follow what the group is doing, as we have funded a number of work in these areas, specifically environmental scientists in aerosol and others. So I'd like to see how they connect with the molecular level type of modeling.
Thank you. Dr. Richard Alo.
Oh, hi, everybody. Yeah, glad to be on board here. I'm Dean of the College of Science and Engineering here at Florida A&M University, Professor of Mathematics, and Computer and Information Science. My areas are computational data-enabled science engineering, artificial intelligence, modeling, simulation, visualization. Recently, 31 institutions were part of a group of 31 institutions that have been funded by Google, LLC, COVID-19, Artificial Intelligence and Data Analysis for Social Good program around the globe. A subgroup of us, 9 of us, are focused on racial and ethnic disparities. In particular, we're working with Dorothy Brown, a research scientist up at Shaw University in Chapel Hill, and University of North research scientist at the medical school there. And here, in particular at FAMU, we are undertaking a transdisciplinary and multi-level study to examine the impact of structural, social, and constructural health determinants, in particular like wealth, racial segregated neighborhoods, food deserts, concentrated poverty, environmental studies, that kind of data, and other determinants of health on the COVID-19 in communities of color. Research is showing that these particular health determinants play an essential role in COVID-19. And so we're really working on to make recommendations to policymakers that they should give some attention to initiatives that protect the health of populations, for example, an upstream approach as opposed to just reducing down to health disparities, rather than solely focusing on providing health and social services. We're also looking at using deep tracing to identify and improve pandemic healthcare, especially where these determinants impact the racial and ethnic disparities that we're finding in the infection and mortality rates. And we're also looking at behavior analytics as identified within social media communications about scientific information and particular public health information.
Thank you. Professor Thaker.
Hi, I'm Julie Thakur. I'm Associate Professor in the Department of Microbiology, Immunology, and Biostats at the University of Rochester. My main interest is in, of course, infectious diseases and connecting innate immune responses to the adaptive immune responses. I have worked with many different viruses, including HIV, RSV, and SARS-CoV-2 in more recent years, of course, more recent months. For the SARS-CoV-2, we are trying to measure the interferon responses and trying to connect those to the clinical symptoms. And from the mathematical side, my group develops models using discrete state models or the logic rule based models of innate immune responses, mostly trying to connect again signaling networks to the clinical symptoms. Yeah, I'm very excited to be here, and I think my goals or my research interests would fit in the physiological models area, trying to look at the host pathogen interactions and the innate immune responses at different levels, intracellular or across cells, innate immune cells. Yeah.
Bruce, we cannot hear you.
Okay, with the technical problem there, perhaps we should just have people, if there are people who haven't spoken yet, you can unmute yourself and go forward. Maybe raise your hand first so we can, if somebody would like to introduce themselves.
Yes.
Until Bruce is back on. Is there anyone who would like to introduce him or herself that has not spoken?
I can. I'm Stefan Hobbs. I'm at University of Virginia at the Biocomplexity Institute. I have experience in multi-scale modeling, combining agent-based models, ODEs or reaction models, which could be also dealt with stochastically, and PDEs applied to immune response in either the gut or the stomach. And furthermore, I've extensively worked in standard development, and I think I can contribute to the modeling technology and partially also on the physiological models.
Thank you. Other people who would like to introduce themselves?
I can do it. I'm Juliano Gianlupi. I'm a PhD student with James Glazier, and I work on multi-scale modeling, usually on the scale of single cell to low thousands of cells. And I'm also a developer of CompuCell 3D.
Okay. Others who were missed? Stocking?
Hi. Yeah, I'm a postdoc at University of New South Wales in Sydney. And the group with and myself mostly work closely with experimentalists trying to understand better the progression of simian immunodeficiency virus in macaques as an animal model of HIV in humans. So we primarily use probability and ODE modeling to understand both viral dynamics and evolution of virus in response to various treatments or immunomodulators. And we also do some work looking at single cell viral production. And there's others in the group who work on malaria. So I would say of the groups you've identified so far, I'd probably put myself in the physiology and viral life cycle sections. Thanks.
Clearly, viral evolution was a topic that should have been on the list that we missed. Thank you for those of you who brought that one up. You're the, I think, third person who's mentioned it. And also there is probably insufficient focus on molecular level modeling. And we didn't want to exclude the molecular dynamics and people working at the molecular scale. Similarly, the people doing airflow modeling and modes of infection barrier function. So going up and down, those are very helpful. So thank you. And next, others who would like to introduce themselves?
Sure. My name is John Bachman. I'm a researcher at Harvard Medical School, and our group develops modeling methods, both for dynamical modeling of intracellular processes, up until now mostly focusing on cell signaling. My personal background is in modeling of apoptosis. And over the last few years, we've been developing tools for automating the assembly of models from knowledge extracted from text mining and databases. I work with Ben Guri on that effort. And we've developed some tools for building up information about causal mechanisms for COVID-19, and have collaborated with the COVID-19 disease maps community, which some of you may be familiar with, and also Corona Y, which is a kind of volunteer effort organized around a coronavirus-related data science. So my personal interests would be on some of the different aspects of molecular modeling. So immune response, host pathogen interactions, and implications for therapeutics. So it spans a couple of those things, and also modeling technology, particularly model credibility, reproducibility, and model integration. Thanks. We're really looking forward to this group.
Others? I realized that
I can go.
Thank you. Amit.
Yes, so I'm Amit Haga. I work at IU, Indiana University. Actually, I'm mainly interested in exercise oncology and the effect of metabolism on immune response. And from there, somehow I got to be interested in bats for a very strange metabolism. And of course, they are the zoonotic host for this virus. And from there, most of my work was doing clinical trials and preclinical trials and integrating that data with CC3D from James Glazier group, trying to use the models to optimize diagnostics, therapeutics, prognostics. So currently I'm interested in using the models to optimize drug therapy for COVID from looking at bats.
Have we missed anyone? Bruce, are you back online?
Can you hear me? Probably a little bit. My Bluetooth system started going out, so.
Well, thank you, everybody. This is just a fantastic set of expertise and skills and diversity that's here. It's just amazing, and thank you for hanging in there for this long. Some people had to drop off, but a whole bunch of people are still here. So I think we probably should not make this go on for much longer. I think our job will now be to propose some organizational scheme for all of this, and this is something we can maybe iterate via some appropriately shared document, via email, maybe via Slack channel. We'll have to figure out sort of what the best way to do that is. But you should be hearing from us pretty quickly, and this video will be available. And so we'll follow up in getting us organized. James, anything else you have to add?
Oh, that's fine. The last call, is there anyone who would like to introduce him or herself who did not have the opportunity? Thank you. I think the action items will be to try to organize the subgroups. Please feel free. We've shared with you a blank document where you can put your thoughts. We also strongly encourage you to take pot shots at the mission statement on the IMAG MSM webpage and critique it. We realize that it was very preliminary. There are a lot of things that could be changed. We encourage you to suggest other people to join the working group because these communities have some overlap but aren't completely known to each other. We also want to, again, the purpose of this working group is coordination, and therefore, if we are duplicating other people's turf, please let us know. That's not the goal. It is to provide a forum for discussion and integration of work and for introductions. And so we will be trying to, I don't think we're going to be having big meetings like this, I hope not. Not from the point of view that I'm saying I'm sorry there were so many people who showed up, but rather that I don't want to tread on your time too much. But I do think that it's remarkable how much expertise and interest there is. Again, this is not limited to COVID. It may have been motivated initially by COVID, but the goal is to think more broadly about what the future holds in terms of host infection, immune response. I don't think there is a silver lining to COVID, but perhaps one thing that will come out of the COVID disaster is that we will take understanding the human immune system seriously. I think that that's a very, very hard problem. And if it can be addressed seriously and solved in some way, that would be remarkable. We have people who've been on the call who are some of the leading people in that area. We appreciate their joining us. We will try to organize some follow-on meetings and have some preliminary subgroup organization. Again, you're free to contact me, Reinhardt, Jim Sluka, and Bruce about any organizational things. I should say that there were almost as equal number of people who were not able to make this meeting time. We were a little overwhelmed by your generosity of time and interest. And we'll do our best to make it interesting for you and worthwhile.
I should actually, James, since we have a lot of people on there still, those of you who still have 5 or 10 minutes, maybe this is an opportunity for anybody to share any sort of general thoughts you might have and things you might want to say to everybody. So in that case, so please go ahead and do so.
Hello. So I have one suggestions that we can formulate, like we can create like an open access Google Doc and start formulating first what are the open problems in this area. I have also been joining a call for a single cell atlas consortium, and the one thing that I learned there is that it's really advantageous to bring a community community, and for that community to bring the most important thing is like first formulating the open problems, and then making a goal for benchmarking some of the known problems that we have that is similar to the open problems. And so in that way we will be able to accelerate some of the new methods, like the development of the new methods, especially because some of the times that I have realized that when developing a new methods, it's very tedious to find out the benchmark those things, like what are the current state of the art. So like to sum it up, we should formulate open problems, how to benchmark existing problems that related to open problems, and what are the state of the arts in this area.
Great, that's an excellent suggestion.
Can I say something?
Please.
Okay, thank you. So there is some overlap with some other groups that are operating even within IMAG. Specifically, I saw Stefan Hoops, he's very active in the model sharing working group, and they actually have a lot to contribute, because if you're talking about integration, that group actually provides the tools for integration. So instead of saying that stepping on each other's toes, maybe it's a good idea to actually reach out to those groups and pull out some of the people there to have an overlap. And just people will be in multiple of those groups.
Yes, the goal of having that listed was to recognize the importance, not to say that we have any ownership of the topic. And so we have issued invitations to everyone who's currently in IMAG MSM and to everyone who's in any of the IMAG MSM working groups.
My suggestion is actually to reach out a bit beyond, because some of the people from IMAG, they have their own local mailing lists. If you get to those, you'll get even international participation in this working group.
Please feel free to send the introductions to us, and we will be sure to follow up. Absolutely.
Thank you.
Anybody else have any thoughts? Well, in that case,
this is John Rice, and I noticed somewhere in the chat somebody talked about setting up a Slack channel, and with the number of people that showed up and the diversity and the depth across that diversity, the communication to start the group up and just really trying to organize who represents what and what they have. The Slack thing may be helpful, and there may be a need for the leadership to develop some short survey questionnaire type formats that are designed to capture specific information, somewhat like what people offered when they did their 45-second announcements. And I've never seen a group do so well holding their time down. But some of those vehicles, because we don't have the opportunity for face-to-face meetings, we don't have sidebars, we don't have breakfast and dinner times. I would just encourage people to be very responsive to short probes that may come out from this committee as people work on trying to refine objectives and interests.
Yeah, absolutely. That's great, John. Communication will be key. And, you know, I don't know that I've ever tried to coordinate 100 people, 100 scientists doing things, but we'll try.
What an amazing briar patch to get thrown into.
You know, Slack is wonderful. I mean, for our coalition, we use that, and it's just been an enabling technology for us, because you can't organize 40, 50, 60, let alone 100 people without its fantastic asynchronous communication. You just get buried in the emails themselves and don't actually get anything done if you work by email. Slack, you can come and go as you need to, and it is scaled very nicely for our community. And you can do it for free if you don't care about archiving all the old messages, which is also nice.
Just need people who have experience like you to volunteer and make your name known to the chairs to kind of help advise on how you do that effectively.
Sure, we can chat with Reinhard and help where we can, but it's beautifully effective.
Great. Any other thoughts by anybody? If not, then thank you so much. This is a wonderful group, and I think if we can just get organized